So this is continuation of the previous lecture. Active transport. The movement occurs against a concentration gradient and requires input of energy. Recall in the passive transport, it occurs along the concentration gradient and does not require input of energy. So then what is the need of active transport when the more economic ways are present? So cost-effective mechanism of passive diffusion, facilitated diffusion are sufficient for transport of many substances, most substances I would say. But sometimes cell has to accumulate a molecule or a metabolite against a concentration gradient or against the electrochemical gradient. And at these places, active transport is required. So यहाँ पर I have shown this you will a positive gradient is being built. This is actually called proton gradient, isn't it? In it, you will learn it about in the oxidative phosphorylation, or maybe you have already done. To move the proton against the concentration gradient, the energy of electron transport chain is used. So this is an example of active transport. Of course, later on, the protons, they move inside through the proton channel, but that is subsequent event. अभी तो आप ये देख लीजिए, this is one example, illustrative example why active transport is required, and so many other substances also. For example, ये lipid bilayer है जैसे extracellular sodium concentration is 137. Intracellular 4.7. So there is a movement of sodium outside. Potassium का क्या है? Oh, I'm sorry, 137, 10. Potassium का उल्टा है. Intracellular concentration is very high, extracellular is low. So to maintain this gradient, it has to be pumped inside. Chloride also you can see. So there are two types, primary active and secondary active, depending on from where the energy comes. Energy is provided by ATP in the primary type. In the secondary, it is the ionic gradient we are going to see which is the source of energy. An example of sodium potassium AT primary transport is this one, which we are going to study in detail. Other examples also, two more examples are there. You can see them later on. So this will require a bit of elaboration. As we said that extracellular sodium concentration is very high compared to intracellular. So sodium has to be moved out. In the first phase, first step, sodium binds with the binding sites. Three sodiums bind. This induces a conformational change. The inside open conformation change to inside to outside open conformation, with the result that sodium also moves out. Release of sodium occurs. Release of sodium followed by binding of potassium. तो क्या होगा? Again change in the conformation and potassium moves inside. So as we had seen that predominant intracellular cation is potassium. By the sequential activities of this pump, you can see 3 sodiums have been moved outside, 2 potassium, and in the process, 1 ATP is utilized. So this is primary active transport moved against concentration gradient using ATP energy. So the ionic pumping activity, there are two features. An inside open conformation which has got high affinity for sodium, outside open conformation that has got high affinity for potassium. Now this part I had omitted. You can also omit it, but just for completion's sake, the जब पोटेशियम बाइंड करता है, potassium binding we said leads to altered conformation, but how? There is sodium induced phosphorylation. Phosphorylate हो गया. Phosphorylation causes change in the conformation followed by release of sodium. जहां पर पोटेशियम बाइंड किया, पोटेशियम इंड्यूस डीफॉस्फोरिलेशन होगा, so the conformation will again change. So stabilization of inside open conformation by dephosphorylation and etc. But don't confuse all this terminology. सिर्फ इसको आप देख लीजिए and try to explain it yourself. So inhibitors of sodium potassium ATPase, a clinical correlate now. These are the two plant sources से मिलने वाली drugs हैं. Inhibit sodium potassium ATPase. Digitalis obtained from Digitalis purpura. This is important for you to learn. So it also inhibits sodium potassium ATPase, and this increases contractile activity of cardiac muscles. This is called cardiotonic effect. So in heart failure cases, this drug is used. So there are two more examples. I have told only one, sodium potassium ATPase. Try to find yourself. One is proton pump and another one is calcium channel. Find out yourself. Calcium channel का you can आपको पता ही है, सार्कोप्लाज्मिक रेटिकुलम, मसल कॉन्ट्रेक्शन होता है तो कैल्शियम मूव आउट होती है. तो ये कैल्शियम फिर अंदर कैसे जाएगी? नो पैसिव डिफ्यूजन, नो फैसिलिटेटेड. तो यहाँ पर कैल्शियम को अगेंस्ट द कॉन्सेंट्रेशन ग्रेडियंट इसको एक्टिव ट्रांसपोर्ट जरूरी होगा. नो पैसिव ट्रांसफ्यूजन पॉसिबल, नो फैसिलिटेटेड ट्रांसफ्यूजन पॉसिबल. Anyway, secondary active transport. ATP is not source of energy. Rather, energy is obtained by coupled movement of an ion. So this is also called ion-driven active transport. So ये you can see here primary and active में फर्क. Only difference is ATP hydrolysis. All right. ATP hydrolysis in secondary active transport, ATP hydrolysis is not there. So we are going to see this part. ये कैसे होता है? This. You see glucose absorption with the help of sodium-linked glucose transporter. Sodium moves along its concentration gradient, and along with that, glucose is also transported. So the mission accomplished, glucose भी हो गया, coupled movement with the sodium. So ये बाद में फिर glucose will move out into the blood. At the moment we are not concerned, just concentrate on this. अब यहाँ क्वेश्चन ये आता है कि ये तो ट्रांसपोर्ट हो गया, एनर्जी फिर कहाँ यूज हुई? तो यहाँ पर सोडियम इट हैज टू बी मूव्ड आउट. ऑल ऑफ यू नो, अभी हमने बात की थी. तो कैसे मूव होगा? विद द हेल्प ऑफ sodium potassium ATPase, and as you have seen, to move out 3 sodium 1 ATP is required. So we can see that in secondary active transport ATP energy is used in an indirect way. प्राइमरी में डायरेक्ट यूज हो रही थी, यहां पर इनडायरेक्टली हो रही है. मिशन accomplished. I said सोडियम तो अंदर आ गया, but to complete the cycle it has to be again thrown out of the cell. तो वो होगा सोडियम पोटेशियम ATPase, जहां पर एनर्जी यूज होती है. Now having completed both these lectures, passive and active, we have come across various examples. कई बारी, जैसे फॉर एग्जांपल ग्लट थे, ग्लूकोज was alone transported. This is called uniport. Antiport, sodium potassium ATPase, ये antiport हो गया, because sodium one direction, potassium opposite direction. और सिम्पोर्ट का एग्जांपल हमने क्या देखा? यही वाला SGLT, ग्लूकोज एंड सोडियम का आपस एक साथ move karna. Now, one thing I have seen repeatedly in Vivas, etc., students tend to confuse facilitated, active, passive with symport, antiport. They tend to say that antiport is a method of active transport. Of course, sodium potassium pumping it is active transport. But I'm just going to give you one more example. ये inner mitochondrial membrane है. So mitochondrial matrix के अंदर all of you know ATP is required, generated. This ATP is required in the cytosol. तो ये ATP बाहर जाएगी where it will be used up and will be forming ADP. Now ADP is required for later generation of ATP. So ADP will move inside. So this is called this transporter ATP ADP translocase. Isn't it a method of antiport? And it does not require any energy. So it is wrong to say that antiport is ATP dependent because we have seen one example where it is not. So we can say it is a facilitated transport because no energy is required. Symport का example देख लिया. Please find out some more example. Build up your knowledge yourself. I am just tempted to refer to this case number 8.1. This is in my textbook, 8.1 case. You just see that clinical case and try to answer yourself. Now last, this is called vesicular transport. We will be discussing in some other lecture, or maybe we will not, because not very important in biochemistry. You will learn it in physiology. Transport of in microbiology also across the cell membrane. You are familiar with this term, exocytosis, endocytosis, pinocytosis. Just be familiar. This is important for you, but this we will be doing later on when we talk about LDL receptors, etc. So this method is called vesicular transport. So we will be discussing some other time.